Well, hello, welcome everyone. Welcome to the NAMA webinar with Dr. Andy Wilson, who is going to speak to us today about why do we make collections, the science of studying mushrooms. And Dr. Wilson is the chair of our Voucher committee, and he is very involved in the Colorado scene. He's the Associate Curator of Mycology at the Denver Botanic Gardens. And we've seen him heavily involved in Telluride, all the NAMA events, the Colorado Mycological Society, I think the Eagle Mushroom Festival probably. He's pretty involved with all the kind of amateur mycology events. I don't think I need to kind of sell you too hard here. I think your reputation speaks for yourself. And I, for one, I'm really excited to hear you talk today about kind of how vouchering has worked for NAMA and how you do it. And I'm really paying attention. I want to hear personally like how people can get involved and become like part of the vouchering effort. And really meet your high standards on doing it correctly. So on that note, I'm gonna turn my video off and sign it over to you. Make sure you unmute yourself there and take it away.
Great. Thank you very much, Trent. Yeah, it's exciting to be here and exciting to talk about this subject, which is, of course, near and dear to my heart, as many of you may well know. And to start things off, Ken has already asked a question in the question and answer section. Why is it called a voucher? Which I think is an amusing question because I haven't really rehearsed the definition of a voucher. But like with anything, like you could ask the question, why do you call a mushroom a mushroom? Where's the name come from? And I think with vouchers specifically, we call it a voucher because it is a representation of that organism that we can use as kind of like a record of that organism in space and time. And the combination of that specimen and where it came from provides us with a voucher or a set of proof for that specimen. And then I think that's essentially getting down to a lot of the topics of this whole issue of why are we making collections? Why do we have a voucher collections project for NAMA and the importance of it? And to sort of drill down on this or just get us started on a mixed message sort of note, we are kind of in a time where we need to start being concerned and be interested in collections as a way of understanding our world, of understanding nature, of understanding diversity, specifically with all the challenges that we will face with a changing climate. And one of the issues that has come up recently is how our institutions in this country and in the world value the efforts of researchers and collections like this. So I'm sure many of you are well aware of Duke's decision to divest from their plant and botanical collections. If you're not, there's a science article that I think goes into it and goes into the mixed emotions and messages about these institutions making these decisions, especially when these institutions have a lot of funding to support the collections. On the flip side, we're in a good point now for fungi. Mushrooms are really popular in the public eye. And it creates us with an opportunity to give messages to the community and to the public about the value of fungi. And one of the ways we can do that are through collections such as the Sam Mitchel Herbarium of Fungi here. And the brainchild of Rick Levy, who's part of our collections team here at Denver Botanic Gardens, he thought of It's a Wonderful Life as being one of his favorite movies and thought, what if we had a message of It's a Wonderful Life, but we told it from the perspective of a fungarium collection. And so with this, our marketing team came up with It's a Wonderful Herbarium of Fungi. And it's a video, it's on Instagram, it's on all of these different sources. And this is, I think, fun for everybody to check out. And what I'm going to be doing throughout the talk here and there is I want to share some of these links and some of these stories with everyone. And I'm going to do it through the chat. So This article from the Duke University closing to It's a Wonderful Herbarium of Fungi through YouTube are going to be in the chat menu in these links. And if you want to tune out for a bit to check out some of these links, go ahead. You will probably also need to backtrack here and there to catch up to some of the missing links. And I'll have these peppered throughout the talk here and there. But for the most part, the talk is going to cover a couple different sections of, so why do we make collections? I also want to go over NAMA's sort of 25 plus years of making voucher collections, a little bit of discussion about how we do it and how the process has evolved over time. And then over the next 25 years, how this process will continue to develop and evolve, and what ways we can anticipate using these collections, why these collections will be important to us. So to start off with, I'm a child of the 80s. I love Indiana Jones. I don't know how my career track ended up being involved or being parallel to Indiana Jones in the sense that he's a collector too, being an anthropologist and collecting antiquities to try to understand history of humanity or whatever he's doing, fighting Nazis, go figure. But we make collections in a lot of ways to preserve evidence of diversity. And in this essence, or in the essence of studying fungi, each fungus is a kind of a treasure in and of itself because of all of the information it contains. And what do we do with collections? Well, not to disparage on the process of sticking the Ark of the Covenant in a box and hiding it away in a warehouse. We do something similar in mycology, although the purpose isn't to lose the specimen. We want the specimen to be stored in a place where it can be preserved over time, but we also want it to keep it organized and in a place where we have access to it. Because you never know downstream where these collections or how these collections are going to become important. And this is evidenced by new technologies, and every time we start learning new things about these specimens, and having these collections tell us about what the species are, where and when they occur, and the combination of all this information tells us something about that organization's contributions to nature and the environment. So having fungaria and these fungal collections are important for us really understanding the value that fungi have to our lives and to nature, and to all the other organisms they interact with. Other groups are also trying to pass along this message. So the Society for the Preservation of Natural History Collections, if you rearrange the acronym, it spells spinach, and that's kind of how we refer to the group. But this is an important group that's very interested in collections broadly: zoological collections, herbaria, plant collections, as well as mycological collections, but also going into like geological, paleontological collections, archaeological collections as well. These are all fall under the umbrella of natural history collections, things you would find in museums, where people can study issues such as the economy, trade, changes of biological organisms over time. And of course, going down the list, you end up on something like scientific treasures, going back to referencing Indiana Jones and the importance and value of collections in and of themselves. And then here's going to be another link I'm going to share, the Society for Preservation of Natural History Collections website. Their next meeting is going to be in Lawrence, Kansas, I think at the end of May. So if anybody's interested in natural history collections, this would be a cool group to get involved with or to look into a little bit more. Also, in some of these places where I have links, there is a QR code. So if you want to pull out your phone, you can scroll on your phone while you have me on in the background. What I also want to talk about next are some of the articles and scientific papers that have discussed the value of fungal collections. And one person I want to point out is Dr. Carrie Andrews. She has put out this really cool paper called Fungarium Specimens, a largely untapped source of global change biology and beyond. And this was in the Philosophical Transactions of Biology. I believe this was dated 2018. And of course, 2018. And she has a great figure one, where figure one, the specimen is Glioporus taxicola. And of course, you can see the specimen label and then some of the handwritten notes about the specimen. And all of these are tied to various different pieces of information you can glean from the specimen, including phenotype information, biochemistry, physical processes, DNA sequence data that tie into other data or analyses downstream, which informs a ton of what we know about these species and these fungi and some very basic fundamental biological questions just from a single specimen. Of course, there's a lot of other interesting points, like what is the cost of not making a good collection? The late Dr. Vicky Funk was at the Smithsonian, and she was a botanist, and she wrote this paper, The Importance of Vouchers, in the journal Taxon, and it was essentially a response by certain scientific journals, American Journal of Botany for one, which were unfortunately downplaying the importance of including voucher information inside of the scientific publications themselves. Kind of a heady subject. It seems sort of trivial, but she wanted to emphasize to the scientific community that you want all of this scientific data. It's very important. Vouchers are critically important to the scientific process. And one of the examples she put in the paper was in the mid 20th century, botanical researchers were counting chromosomes. And they were getting chromosome counts for a number of different species of plants. And this is important because if anybody studies plants, they know that different plant species can actually be differentiated by different numbers of chromosomes that you would find in their cells. Now, there were thousands of specimens that were examined, leading up to thousands of chromosome count numbers, but none of these specimens were saved. None of them were vouchered. So, in essence, what you ultimately had was an expensive and arduous list of numbers that could not be back referenced to any kind of biological data beyond just those numbers. And that's a challenge. That's a problem. If you can't connect these numbers back to any sort of biological or taxonomic species, then you essentially spent a lot of time and energy on very little value. And that's where the value of the specimens comes in. Another important area comes in. Dr. Nilsson, who's in Sweden, and he's an extremely prolific researcher, largely works with DNA sequence data. And he was back in the early 2000s looking at DNA sequence data on commonly accessed databases. Now, again, this is almost a 20-year-old paper, but these numbers haven't changed, specifically in the very general aspects of GenBank and the reference sequence databases that we use. About 27%, nearly a third of all ITS sequence data comes up being recognized as insufficiently identified. To double down on that problem is that 3/4 of these insufficiently identified taxa can't be cross-referenced, or sorry, I said taxa, but sequences can't be back-referenced to known species, right? So you've got known DNA sequence data, which represent good sequences that have good reference material, and we do know the taxa that this DNA sequence data attaches to. But then this insufficiently unidentified sequences cannot be matched, or at least 3/4 of these do not match anything on this record. So essentially over here in these insufficiently identified taxa or sequences, you have a black hole. Now, one solution to this would be if we had vouchers for all of these sequences, we could then drill down on the specimen and get a good identification for those specimens. And he says here, with no specimens to support the sequence, how can you validate the sequence itself? And this follows up with a more recent paper by Dr. Robert Luking, who used to be at the Field Museum in Chicago, but is now back in his home country of Germany. And he has a couple very important things to say, especially on this topic, unambiguous identification of fungi. Where do we stand when it comes to fungal DNA barcoding? And it's a pretty lengthy paper, but it's a great review paper because it covers a lot of territory when it comes to understanding how good DNA sequence data is in giving us an understanding of diversity. And in here, he points out the reference data is the bread and butter of identification tools. Where there are no keys available, it is necessary to consult published descriptions and reference specimens. It's often painstaking yet indispensable approach to how that is nowadays facilitated by digital repositories and databases, which I'll talk in a little bit more later. The accessibility of reference materials such as vouchers, both physically and virtually, is critical to this process. And one of the things he talks about is more recently, there's been the development of a highly curated reference database in GenBank, as well as Unite, that helps connect the DNA sequence data to actual physical specimens. So we do have validations for all of these taxa that are in this database. Now, one of the challenges, though, is that these reference databases, such as RefSeq in GenBank, that have highly curated DNA sequence data, they're incomplete. And this is kind of pointed out in one of my first graduate students' research projects, where we developed a modified sequencing method to try to get mass sequence data from Fungarium collections. And one of the tasks I asked him to do was, okay, he's got over 700 DNA sequences from all of these different collections here at the Denver Botanic Gardens. What is the chances that this DNA sequence from this specimen matches a sequence from a known database? And here you've got a couple different examples of different databases. You've got the generic GenBank nucleotide database, which has all of the ITS sequences in it, whether or not they're sufficiently or insufficiently identified, as Henrik Nielsen's paper pointed out. Or you've got the highly curated RefSeq database. Now, this is going to have a lot more sequence data in it, but they're not well-known taxa. This is going to have fewer DNA sequences in it, but all of that DNA sequence data ties back to a known species, right? So this is highly curated. This is less highly curated. Well, when he tested all of these hundreds of different species or specimens, what he finds is that there's generally good DNA blast matches to the reference or the general nucleotide database, in the sense that you get upwards of 97.5 sequence hits to a lot of these specimens. But how many of those sequences are from insufficiently identified taxa? You look at the RefSeq database, we find out that there's a number of taxa that we have in our collections here that aren't well represented on the RefSeq database. So this shows that we can actually use collections to help flesh out some of these reference databases and get better at using DNA sequence data to help identify fungi downstream. But of course, it takes a lot of work. Another cool thing that comes out of the paper of Robert Lukings is also pointing out the fact that the barcode for fungi is imperfect. There's a lot of nuance to using DNA sequence data to help define taxa, and species. And this last year, with another group of mycologists, I published this paper. And I'm going to drill down on it a little bit, but not try to overwhelm people too much with the nuances of this paper. It's actually somewhat a simple concept, but there's a lot of complications going on in it, if you can sort of bear with me for this. What we wanted to understand is, does one size fit all? Does the variations in the barcode gaps of macrofungal genera prohibit some of their ability to use the ITS as the fungal barcode, or muddy the waters? And what we mean by the barcode gap is understanding the difference between intraspecific distributions and interspecific distributions when looking at pairwise distances. What that is to say is, if I get the DNA sequence data from one mushroom and the DNA sequence data from another mushroom, I can do a pairwise comparison of that DNA sequence data. The DNA sequence data from things that are the same is an intraspecific comparison, such as here. So two species are the same. They're going to have very similar DNA sequence data. As a result, the pairwise distance between these scores is going to be low. It's going to be to the left of this x-axis, and the distribution is going to be somewhat narrow. Okay, so if I have hundreds of different comparisons of individuals that are the same species, the pairwise distance numbers is going to be small because they're the same species, right? You want the DNA sequences are going to be the same. If I do an interspecific comparison, that is, two different species are being compared, you're going to have a fair amount of variation between the DNA sequence data in that pairwise comparison. So if I have hundreds, if not thousands, of pairwise comparisons, the numbers will be larger, further to the right on the x-axis, but there's going to be a lot more variation. There's going to be a lot more spread in this distribution. So hopefully that kind of makes sense. Based on this theory, we want to see how does this barcode gap, the difference between the interspecific distribution and the interspecific distribution ideally has this chasm between these two distributions, and this is called the barcode gap. But sometimes there might be an overlap of these pairwise distances, and that's going to create a central problem when you're recognizing same species versus different species. And if this is an issue with looking at the ITS sequence data, then how much can we rely on that sequence data as a magic bullet or a silver bullet in identifying fungal species? Now here, this is just a few of the genera we looked at. I think we looked at a total of at least 9, 10, maybe 10 different macrofungal genera. We have the genus Sewillus, Russula, and Styrium in this example. I have bar and whisker plots because it just allowed for examining the distributions a little bit better. But if you can imagine, the distributions are on their side. This is the scale of pairwise distances on the y-axis instead. Here you have the interspecific distribution and the intraspecific distribution for Sewillus of the entire ITS region. And you can see, maybe if you look at this versus Russula versus Styrium, there might be in some cases where you do have overlap of these pairwise distance tails and the loss of the barcode gap. So how does this happen? I also have a little illustration as to whether or not you're treating your populations. If you're lumping species together, like all of these things look the same, we're going to treat all of these specimens and individuals as the same species versus splitting. Actually, we're going to break up these groups into this taxonomic species and this taxonomic species. These can have an effect on whether or not your distributions versus intraspecific and interspecific comparisons are wider or narrower as a result of this lumping and splitting. Anyways, there's nuances to understanding these questions and using ITS sequence data alone, which is why we want to be able to always revert back to something like a specimen and have a specimen there for us to maybe look at the morphology, look at other features, and generate more DNA sequence data from other markers beyond ITS. Having a vouchered specimen gives us room to understand these organisms much more thoroughly. So the Voucher Collections Project was made over 25 years ago for NAMA, largely because of this issue of wanting to understand diversity within North America better. And there's a history behind this. I mean, of course, NAMA goes back many, many decades, so probably over half a century. And there's lots of very important people. There's Orson Miller, if you can recognize him as a young man. Here's another picture of Orson Miller. In the more recent years, there's Dr. Harry Tiers, the father of Dr. Barbara Tiers, and Hope Miller is there, the great Eileen Stanley, and as well as Ben Wu. Very great members, both professional and amateur mycologists, have been mingling at NAMA events for years and years and years. 7. All of the diversity information from these four is essentially a list of names. Ultimately, Eileen Stanley, who you can see here, who became president in the early 90s, I think she dawned on her and she became a little bit distraught back in the early 90s, seeing all of the display specimens get tossed out into the garbage. Because it dawned on her is like, well, don't these things have more value than simply just being refuse? And the answer ultimately is yes. I mean, she knew the answer at the time. And I think at that point, sat down for lunch in like 1995 with Dr. Greg Mueller, and they hatched out a scheme. Let's start vouchering representatives of all the different species we put on our species list. These can be stored in a museum and they can be used for back references to help validate these taxa and maybe even better understand this diversity, which are probably new species in one way, shape, or form. So in 1997, ended up being the first year of the Voucher Collections Project, and this was ended up being headed by Dr. Jack Murphy, who did all the vouchering for that first year. He was later, this role was later subsumed by Dr. Patrick Leacock in 1998, who ran the vouchering collection project for more than 20 years, and created a lot of the organization and the system that we see today that I will share with you part of this project. Patrick also runs his MycoGuide website, which has a great, it has a great history of NAMA and NAMA's data. I'm going to create the link for that here in the presentation. And essentially, here he's got a species accumulation curve. Now, this is an accumulation curve of new species that come in through each of the forays. Now, this goes back to all the way to 62, some of the first forays and lists. But of course, you have to go all the way to 97 before you actually have vouchers to validate any of the species that occurred after it. All of this before is kind of like what happened with that chromosome count number. We have species names on a list. We have no way of validating those species names. But since 97, and since we vouchered these specimens, we can now go back, look up those specimens, and make sure the species name we applied to this actually works. We can even go back and get DNA sequence data. I'm going to stop here for a second and pause because Joe has a question. Are ITS sequences with no vouchered specimen still potentially valuable to science if they match a vouchered specimen? I think this would still provide valuable biodiversity data, especially if there is an INAT or other digital observation. Thoughts? I agree. I mean, it's not to say that if you have a DNA sequence DNA sequence, and no voucher, that the DNA sequence lacks appropriate value, but there comes in a number of questions and risks of not, as I mentioned before, of not having the voucher. What if you fail to make a collection for a specimen and the DNA sequence data is something that we haven't seen before, or matches DNA sequence data for a specimen that we've been on the lookout for a species we wanted to make more collections of? DNA sequence data helps make us more aware of these potential issues. But I think it also makes us more aware of the need to try to voucher a lot of these collections and ensure that these specimens are available so that if we do learn something new from the DNA sequence data or even the iNaturalist observation, we have the specimen to back this up. Now, one of the challenges is that if you have an iNaturalist observation and a DNA sequence and no voucher, you might say, oh, this is a new species. This is something very new and exciting. And you might come up with researchers that are in agreement with you. They're thinking this is a new species. We can describe a new specimen. The scientific method and the rules around describing new species require that a voucher exists for that species. If you don't have a voucher for any of these specimens, regardless of the number of observations or DNA sequences, you can't describe the new species. There's not going to be any scientific publications out there that are going to accept your work without a validated and verified voucher. So yet another reason why it's important to ensure that we make these vouchers, because downstream, you never know how many new species exist in collections like here at Denver Botanic Gardens. We actually do know of some. Graduate student Justin is probably going to be describing new species in his research work, largely based on specimens here. So, good question. Thank you for asking that, Joe. Happy to follow up on that in any future discussion. Now, with the diversity of NAMA specimens, we've got issues such as we've got the accumulation of taxa and specimens over time. Now, this sort of record, this accumulation curve is just for the specimens themselves. Now, this collection only goes to 2019. And of course, since then, we've gotten several hundreds more specimens added to the collection. So right now we have well over 8,000 specimens total in the collections that are currently at the Field Museum in Chicago. Now, these are specimens. What about the number of species? I've since created a species accumulation curve from all of our taxonomic specimens, and this number goes back to last year in North Carolina. Normally, when you see a species accumulation curve, if you start saturating the curve and saturating the amount of diversity, what you'll see is a parabolic sort of curve, and it will level off at some point because you've sampled all of the diversity there to sample. But of course, over the years, this number has been steadily climbing. We're finding new species at each foray every time we make vouchered collections. And of course, because North Carolina was such a boon, we had over 500 specimens being vouchered. You see a nice little uptick in the amount of diversity that is found there. And right now, that crosses over the threshold of 3,000 species documented over the last 25 plus years for the North American Mycoflora or North American Mycological Association's Voucher Collections Project. This diversity represents a lot of different taxa. These are all the different genera, and this represents the top 50 voucher genera from the project. Russula and Russulaceae is, of course, the most diverse groups right now, including Amanita and Cortinarius. Russula has the most number of specimens overall. However, it only has the second number of total species next to Cortinarius, which has the fourth number of total specimens overall. As far as like the total collection's diversity, we have more agaricales than anything, right? And then of course, Russulaceae are in there quite a bit. What I'm mostly interested in, I want to see more of is Thelephorales taxa. Given what we know with environmental sequencing data, Thelephorales are the most diverse group of fungi living in our soil environments on this planet. There's a ton of diversity that's out there. The problem is they're the little crust fungi. There's these little genus Thelephora. We get some Hydnellums and other taxa in there as well, but there's a ton of these Tomentella-like little crust fungi out there, and they are the most diverse genus in soils on the planet based off of a recent environmental sequencing study from Eastern European groups or Eastern European researchers. Another thing I'd like to see more of is we can need to collect more ascomyces. There's a ton of cup fungi out there. The only thing is that people need to get on their hands and knees and start turning over logs and turning over these things. And of course, I think one of the issues would be is getting more ascomycete taxonomists to our forays, because I feel like we're getting some ascomycetes at the forays, but we don't have enough people to help us identify these fungi and as well as collect them. Of course, what's also important are the people. There's a ton of people that are out there collecting. We have a number of shared contributions from both academic as well as amateur mycologists. So all of the values here that are in yellow are academics, actual practicing mycologists who do it for their career, and as well as all of us and the rest of you guys who do this for fun. We have tons of contributions in terms of total collections. And at the NAMA forays, of course, 91% of all of our collections come from the amateur community, less than 10% from the professionals. When it comes to identifying the taxa, we have tons of contributions from the amateur community as well. I don't know why Walt Sturgeon. I want to say Walt Sturgeon's professional. I mean, I've seen him enough around to say like he's got to be a professional, but for some reason this has been colored blue. Somebody else can correct me on this. But these people represent the backbone of everything that we do. And there's a ton of contributions from professionals out here. I got some great pictures. We got, of course, Dr. Jesse Glazier at the Namaphore in Idaho in 2008. And here working at the microscopes, helping ID things along with Dr. Jean Lodge, Dr. Kathy Cripps from Montana, Adele, our venerated recorder, and of course, my predecessor, Vera Evenson here at Denver Botanic Gardens, helping things out. I want to say that this is Dr. Michael Bugh's back. It looks a lot like the back of his head. I need to get him to come in and confirm whether or not that is him. Of course, we have other contributions by Steve Trudell, Debbie Weiss, Dr. Elsa Valinga, and Dr. David McLaughlin, along with Dr. Maj Padami. The late great Tom Volk, whom we all miss, is here with his graduate student Matt Foltz, likely looking at some sort of species of chanterelle. Everybody knows Anna from the Minnesota Club. Hopefully, Minnesota's in the crowd representing. Dr. Elsa Valinga again, as well as everybody knows John Plischke. He's definitely involved in the DNA barcoding aspect of things going on nowadays. Jeff Kibbe and Ray Fatto are likely trying to confuse J. Justice with this species of Russula, but of course, a ton of what we know about North American Russula comes from these two gentlemen on either side. Clark Overbo, also a great taxonomist, along with Texas's own David Lewis. All these people are very important and contributing to all this diversity over the years. Now, Glenn also asked a question, what about microfungi? I think this was in follow-up when talking about vouchering things. There are repositories for microfungi. And I think when we talk about microfungi, we can talk about anything about when it comes to like yeast, single-celled fungi, or filamentous fungi. There's going to be some challenges because in order to store and preserve microfungi, you want to be able to have some kind of voucher like culture. But in some cases with microfungi, what might end up happening is that the microfungi are described based off of permanent slides. Like microscope slides can be made where you can look at these microfungi under a microscope and you can preserve that microscopic morphological characteristics under these things like permanent slides. Those are then what end up getting vouchered. But microfungi are a sticky wicket, and there's a lot of diversity that has been described in a number of species that have been described. And again, because so much of mycology falls under the botanical / fungal code of taxonomic nomenclature, there's still this rule: we need to have some kind of material or voucher for those specimens. Now, vouchers can be more than just specimens. Specimens are ideal. There are vouchers that are simply paintings, that are simply drawings. You can go to a lot of these old museums and look up a voucher for a particular species and a particular type collection, which is the one voucher that describes that species over all vouchers. And it's just simply like a line drawing that some researcher came up or drew in the 1600s. Since then, there are other vouchers that represent physical specimens, but they refer back to that type specimen or that drawing. So there's a lot that can go into creating a voucher and then using that voucher for the scientific record. So, but good question. Vouchering isn't always easy, depending upon the specimens. Okay, so trying to get through some of this stuff, I've been taking up a good fair amount of time. Yeah, so how do we do it? How do we do this voucher collections process? And here I want to give a shout out to NAMA and all of the support that they provide to us who make up the core of the voucher collections project. I'm talking about Liv Filialuna, Patrick Leacock, and Wyatt Gaswick, who's also at the Field Museum, or their contact point at the Field Museum. They give us so much support in bringing in all of these amateur mycologists as well as students to help us do all of the work that we do at these forays, because there's quite a bit of it that has to happen. And how we do it has actually been documented more recently. There's a couple of different articles that have and what do you call that? So like Instagram articles and stories that have shared our process and what happens. And part of it is a lot of it goes to Liv Filialuna and their contributions. Liv created this awesome chart for us that is in this article that we co-authored with uh, Wyatt Gaswick and Patrick Leacock. And this is in the Mycofile Quarterly of this year. It's the very first one. If you want to go ahead and check that out, I'll include the link in a second. And of course, as I mentioned, there's the Instagram article that Liv put together. There's a couple different articles, and they're very well done. They did an awesome job with it. And of course, before I've mentioned Patrick's website, he's actually got a section on there on the history of the project or the voucher collections project, as well as some PDF documents that sort of illustrate and describe our process. Here are some of the links inbound. Several of them are Instagram links, so you can check those out as well. And basically, the process is fairly streamlined. Essentially, what we try to do is we want to have a way of organizing and capturing as much diversity that come in from the field as possible. So these field collections come in from the foray, and they end up showing up in wherever the display area is. And we first try to funnel the members who we treat like cattle, and we get them to their foray tables, and we have them to go through sorting and labeling. Don't want to treat the process as inhuman, but we have so much going on. We kind of have to sort of like get these things processed in an adequate way. And what is important is that we have these specimens with good labels, because if the specimens don't have good location, date information, as well as the collector information, the value of that specimen as a potential voucher is significantly reduced. We're not going to take up space in a museum if we don't know where that specimen came from, if we don't know when it was collected. So because we need all of this data, we want to make sure that people are filling out their slips, and we also coordinate them based on their forays. So that helps us get the where information. And of course, if we know which foray it came from, we know what date that foray occurred on. So a lot of this stuff can be really well managed if we just make sure that everybody gets the foray information on their slips, as well as their personal information and ecological information, if they can, onto those slips as well. The next step is that we use whatever information was provided, including environmental data, as well as looking at the specimen itself to undergo identification. And sometimes this can be a little bit arduous. Here's Britt Bunyard trying to identify this Amanita. He's actually looking at his own book. And I love this picture because I caught him just right at the moment where he was clearly rubbing his head, looking confused, but he knows what he's doing. Of course, you have great and venerated mycologists come through who can help us identify things, just like Ellen and Arlene Bassett, as well as Bill Yule here in North Carolina, helping us break records in terms of total numbers of species vouchered. The identification process is the critical step for everything that we do, just to make sure that we're capturing as much diversity as possible. It's usually one of the bottlenecks too. Once we've identified specimens, this is where we start the processing in the real vouchering, or at least the recording of the data that goes in for each of the vouchers. Now, each of these vouchering specimens kind of gets stuck in like a holding pattern as Adele goes through and checks and makes sure that she's got all the information for each specimen, not just simply like looking on her phone. So I caught on this image. They then, once they get vouchered and their information goes into the database, other members of the group like Wyatt Gaswick will take ex situ images, which means you're taking an image of the fresh specimen but outside of its environment. iNaturalist is great because it helps you take in situ images. This is our ex situ imaging process. So we at least capture some of that morphological data from the fresh specimen. And once it goes through this process, we then decide whether or not it goes on display or it gets dried. and processed for vouchering, and sent to whatever collections place we need it to go to. And the display specimens, of course, are the bread and butter of the display area and a lot of the foray, where people get a chance to sit down and look at all of this diversity and learn what the species are. Here, Bill Yule is holding court and instructing about the volatales here. Here in Colorado, you've got Clarissa, as well as Justin Laux and Patrick Leacock, organizing and sorting some of the specimens that come through the collections. And then lastly, of course, the specimens need to be housed somewhere. And the Field Museum in Chicago is our main repository for vouchering these specimens. Of course, we need to make sure they all get dried while they're at the foray. If not, they need to go to some sort of like local institution here. After the Colorado NAMA foray, they went back to the lab here at Denver Botanic Gardens, where the rest of them were processed and dried. And ultimately, they need to be packaged, sent to the Field Museum, where Wyatt Gaswick and Patrick Leacock help finish the processing of the specimens. Now we're also evolving in the sense that we're taking on new ways of getting data. So such as tissue sampling for DNA is something that is sort of new, that is developed over the years. And each progressive foray since, COVID and maybe even pre-COVID, DNA has become more integrated into the process, and figuring out how to make this process a little bit more seamless and capture the DNA sequence data so that they get connected to the vouchering process is something we're looking into. And over the next 25 years, we're going to be adopting new methods, and we're going to be coordinating with all sorts of different organizations to make sure that we're doing our best. I actually just realized I need to update this MSA because the MSA has updated their logo. So sorry, MSA, if you're in the audience. But anyway, there's lots of new tools that have come out since the voucher collection project started. iNaturalist is a huge increase into the value of the data that we can attach to our vouchers. And now Nathan Wilson, and I'll talk about this a little bit later, Mushroom Observer was the iNaturalist for mushrooms before iNaturalist even existed. And it's been around several decades, probably a good decade. I can't remember when it first started, but it's potentially a decade before iNaturalist even came onto the scene. Now Mushroom Observer is starting to develop their app, and they're trying to make their tools a lot more useful for vouchering and using as we collect mushrooms and document mushrooms more broadly. It can be interact with iNaturalist, so you can connect observations between the two tools, and downstream it might be even more useful in the way that it helps us record data and what Adele does as the recorder. But in this process, we incorporate vouchers because they're important links to this whole process. Right now, you've got workflows where people can go into nature, create and make an iNaturalist observation, extract DNA from that specimen, and create a DNA barcode. There you've got some data that allows you to look at things such as iNaturalist will allow you to look at morphology to a degree. The DNA data will allow you to connect something like population information, some omics information. You can get some good DNA data out of that, that connection and that workflow. But when you have a voucher, you get so much more in terms of like versatility of the information you have. You can do all of the same things, but you can get the omics and population genetics, but you can also do something like, especially with fungi, look at chemical information, look at dyes. There's a paper that came out recently looking at the taxonomy of Cortinaria species and the dyes that they produce. You can't do that without specimens. You can't look at isotopic data if all you have is the iNaturalist observation and DNA. That voucher helps create the link, and it's a sort of a trifecta in this informational list or informational gathering that we've got. Now, the problems are, how do you voucher? Not the average everyday person doesn't get to voucher their specimens because there's barriers from taking the specimen and getting it into an institution like Denver Botanic Gardens here. Now, fortunately, we've got these new tools. We've got online tools like iNaturalist where people can create that information from the vouchers. That information can then be put into the reference for the specimen in the online database for specimens, which is the Mycology Collections Portal, or known as the Myco Portal. But there's so much that is undefined to most people about this process. And sometimes the institutions like mine tend to be this angelic space in the clouds where it's hard to access. Nobody gets access to it. One of my roles and one of the things I want to try to do is demystify this process and work with groups like NAMA so that institutions like Denver Botanic Gardens or even Harvard's Farlow Herbaria aren't inaccessible to the average collector. And there's so much of the good work that the amateur and clubs and all these people are doing to help us document these fungi. This is not to say that the process isn't complicated. There are a number of different steps, from everything from drying the specimen to properly labeling it, to making sure the species identification is good and recording all of that data and making sure the specimen is properly processed so it can ultimately have a home in the collections. Now, we are working with groups like the Fungal Diversity Survey, with NAMA, and I'm also working with professional clubs or professional institutions that house these collections to figure out how we can get everybody on the same page. I'll talk a little bit more about why this is important and how institutions are recognizing the importance of collections from groups like NAMA. So we want to increase the opportunities for the mycological community broadly, not just professionals, but all groups, to contribute to this vouchering process. One of the ways is, like I mentioned, is that Nathan Wilson is actually actively trying to develop the Mushroom Observer app. And we even have a meeting tomorrow to figure out how Mushroom Observer or NAMA in and of itself can improve our databasing of specimens from forays so we can have a more streamlined approach of getting the voucher specimen into this, the collections, but also linking this DNA sequence data with the voucher specimens more seamlessly. There's already processes in place, but a lot of these protocols and steps are very disparate. Mushroom Observer can be the one-stop shopping place for doing all of this. I think that's going to make a lot of people's lives much more easier and link people through the vouchering process much better. I want to bring up something that I've been working on. And last year in 2023, I started bringing together a group of individuals from these professional fungaria to try to understand the needs and the requirements of fungaria, considering the issues that Duke had and what many collections and museum spaces are having with preserving specimens. Like, how can we get more resources and or instructions to your average everyday collections? Because even though an herbarium or a museum might have a fungal collection, that doesn't mean they have the expertise to deal with it, because museums can't fund tons of different people. A lot of these institutions are underfunded, and they might only have a botanist in their institution. They might have a huge fungal collection, but they can't afford a curator or a manager of those collections. So what I wanted to do is sort of create a support group for all of these different kinds of researchers and collectors to see what we can do to maybe help all of these other institutions and ensure the value of their collections going forward. So this is the Fungaria Management Working Group. There's 26 plus contributors from all over North America, and even including Lee Davies, who's from Kew Royal Botanic Gardens, and we're probably going to talk about publishing some resources, including a Fungarium handbook, so people know how to handle specimen collections. And one of the things that we did towards the end of 2023 is talk about making a survey. And so in late May of this year, we created a survey that went out to 300 plus North American institutions that we mostly believed had fungal collections, and asked them to contribute some answers and answer some questions that we had about their collections and what they thought about the future of their collections. So far, we got like over 90 different respondents. And from those 90 different respondents, I had two different results that I thought were good. Um, Question number 10 from the survey was, what area of fungaria management is needed, desired, or most important to you as a collector and part of the collections? And so the top answer was that they felt they needed more taxonomic expertise. And this is probably something where the mycological community can come in and help, because there's a lot of great identifiers. As I mentioned in the slide before, some of our top identifiers at Forays are the amateur members of the amateur community. So if you're really geeky about mushrooms, consider upping your taxonomic expertise, because these collections could really use your help. Other things such as digitization of records, learning about best practices resources, these are all important. One of my favorite responses from some of these survey questions is on this next slide. Of the groups listed below, please rank from 1 to 6, most to least, which contributes the most to collections. Basically, who is adding to their collections most of all? Just behind the professional mycologists were the mushroom clubs. You guys are the most important to contributing to these collections from these respondents' points of view. Now, this is important because I want to work on the future is how can we get more of you working with these collections? How can we provide more of you with access so we can scratch that geeky itch you might have of becoming more fluent with fungal taxonomy? Because we need more knowledge about mushroom diversity out there. One of the important things about these collections too is how important they might be to our understanding of conservation going forward. Over the next 25 years, and something that's happening in Colorado is that Dr. Amy Honan and I have approached the Colorado Natural Heritage Program. You probably heard of the Fungal Diversity Survey has been working with the California Institute for Biodiversity, and the California Institute of Biodiversity, which is a natural heritage program in California, they've got millions of dollars and they've been throwing tons of money towards understanding fungal diversity in California. Amy and I were thinking, well, surely there's got to be something similar in Colorado. Well, it turns out the Colorado Natural Heritage Program got over $8 million to update their understanding of rare species and endangered species in Colorado, letting us know that they're the group that we probably need to talk to if we want to start looking at conservation of macrofungi writ large. So there are plenty of natural heritage programs across the country. Every state has their own natural heritage program or project going on, and they all kind of contribute to the Nature Serve, which is sort of like a database for sort of understanding rarity and endangered taxa across North America. The challenge is that these groups have been focusing mostly on animals and mostly on plants. They might focus on some other weird organisms here and there, but that's been their primary focus. And there's really very little reason why they can't include macrofungi. And so when we met with the director of the Colorado Natural Heritage Program, David Anderson, he was ecstatic that we came to him and that we're going to start working on including macrofungi as a group that needs to be considered in terms of working on conservation from a national perspective. We're starting locally with here in Colorado, and we recruited Dr. Scott Bates, who is one of the principal developers of the Mycology Collections Portal. And through the Mycology Collections Portal, we can actually use specimen information to understand species rarity in Colorado. And he's working on that. And we're also working with James Shaleen from the Pikes Peak Mycological Society to start off with a species list of rare fungi that we can start sort of monitoring. And then also sort of create a framework in which other states can really take what Scott is building, apply it to their own state, and start looking at what species that we might want to start monitoring and looking into for conservation purposes. So there's so much that vouchers can offer, and I'm really happy to give this presentation to all of you. And I want to acknowledge my co-conspirators, Dr. Patrick Leacock, there's Wyatt Gaswick and Liv Filia Luna. They're integral core of our voucher collections program. For a lot of our photos, going back into the past, Dr. Michael Bew, Dr. Kathy Griff, Dr. Gray Miller, and David Lewis have all been instrumental in providing a lot of these images. And I want to thank a lot of these contributors for our committee, as well as external liaisons, and a lot of these institutions that really have been supporting a lot of the work that we do. And so with that, I want to thank you. And if there is any more questions, I'm happy to answer them. And does anybody have any thoughts? I think, there's a couple of questions here. So Joe said, we'll follow up later offline. Could specimen vouchers be collected for a previously sequenced specimen and be used in describing a species? Or is the rule that all of your sequence data has to come from a physical specimen voucher? No, there's no hard and fast rule, although if you want to say something important biologically about the species that you got DNA sequence data for, and you don't have a specimen of that species, you're very limited into what sort of biological questions you can answer. Now, it's important to sort of say, okay, I've got a DNA sequence here for the specimen that I collected at this place. It actually matches the DNA sequence for the specimen that I do have a voucher for. And through there, you can start making connections about that species. The only problem is that these specimens might have been collected in, say, Montana, but this one that you're curious about, you collected here in Colorado, and you've got no more specimens from Colorado. So next time, what you might want to do is keep an eye out for it again, and then get vouchers from that place. So it might be enough to say you got species, representatives of those species, but it's even more to say you've got representative of those species in place and in time. And in many cases, sometimes if you just record DNA sequence data, like so much of GenBank has, you have inadequate information about space and time for that DNA sequence. So you're kind of limited about some of the biological questions you might want to explore. Yeah. Any further thoughts, updates on the Duke University Herbarium Fungarium? Not quite sure about what the updates are. I feel like, I'm talking with Barbara Tiers, who is, I think, one of the central figures in herbaria, and not just North America, but in the world. And she's actually volunteers here at Denver Botanic Gardens. And she's been deeply involved in this question of, well, okay, well, what are we going to do with these Duke specimens? That the plan or the hope is that there can be a co-investment between Duke and the University of North Carolina, which is nearby in Chapel Hill. I think it's Chapel Hill, I don't know. But they're two universities that are very close together. And if there is an agreement that comes between the universities where they can share investments, they can potentially move those collections not too far. Because even though it's like millions of specimens, a lot of it is very important representation of botanical diversity in one of the most biologically or botanically diverse areas of the country, if not the world, which is the Smoky Mountains. There's so much incredible diversity there, and many of that is housed at the Duke University Herbarium. And it would really suck if that regional diversity ends up having to go to a completely different state or part of the country just to preserve it. So hopefully there's some effort within the herbarium community to move these collections to a facility that's shared and that it's regional. But it's a developing process because they only learned this year, and it's probably going to take years before they can figure out what the next steps will be. And it's going to be a challenge given, everything else that's going on in this country. Molly in the question and answer section says, what's the best way for those of us from small museums to access resources, connect with folks from the working group? I'm not finding much with a quick online search because we don't have much of an online presence. That's why we've been very kind of insular. And I think the only way we've been able to reach out to people is through some of the emails we sent out, and that we gleaned off of Index Herbarium, which is a database of all of the herbaria in the world. And so we tried to contact and reach out to people broadly, and that's how they might know of us and be involved with us. But Molly, if you want to reach out to me, I'm at andrew.wilson@botanicgardens.org. Go ahead and send me an email sometime, and I can try to make sure that you're on whatever distribution list that we have for communicating more broadly. I'm feeling like in 2025, what we should be doing is having like maybe semi-annual newsletter for the Fungaria Management Working Group. If and when this translates into more of an online presence, such as a website, that might happen downstream. Liv Filialuna, who's part of my NAMA team, is also part of that Voucher collections team. So they might be able to help with our online presence in future discussion. I would also encourage you, Molly, if you want to attend the MSA meetings in Wisconsin, in Madison, Wisconsin next year, because we will have a symposium on fungaria and voucher collections. This is a lot of this is in response to that of the whole Duke kerfuffle. So yeah, that's that. And I think, what else, George? And then George has a question in the chat. Let's see. I want to make sure, and I don't know if I answered your questions. Sorry, George, hold on, I'll get to you. There are some interest in having next year's vouchers out of Right in the Rain paper. And there's just a possible future chemical analysis. Is there a minimum amount of species tissue needed? Oh, I don't know that. I don't know what I'm sorry, I'm not sure what that first question is in reference to. But yeah, I don't know. The minimum amount of species tissue needed, there is, like I mentioned, there is probably like a minimum amount, but there's also a maximum amount. It depends upon the method, the chemical analysis that's being involved. And I'm sure what you probably need to do is just find the recipe, the essential protocols for running those chemical analyses. But usually, I don't think it's a ton of material, depending upon the method. So probably just less than an ounce, probably just like maybe even half a gram or something like that in some chemical cases. So going to George's question, are there any video module with clear instructions on how to collect and where species or where expected grades you're looking for? Dr. John Rubin at San Diego Natural History Museum trains volunteers and amateurs on how to collect data on INET. Okay, that's a great question. So what I can do is I'm going to post in the, I can find where my YouTube channel is. Actually, there was a couple years ago, I teamed up with Gabriella Delia on essentially on how to collect macrofungi for research, and we have a YouTube video. Yeah, here you go. Hopefully, let me get this shared and in the chat. And what you can do is reference this. This is essentially a video that Gabriella Delia and Gabriella produced, but it gives you sort of like a step-by-step instructions on how to ideally create a macrofungal specimen for vouchering and how to try to collect that data and what's the data that you want to collect. Because there's a couple of different steps. You don't really want to just like rip a mushroom out of the ground. You also want to incorporate some information about the habitat that it's in. But you also, in some cases, you might, if you ever had this experience before, you might open up a specimen and it's chock full of dirt because somebody collected more substrate than mushroom. And then somebody else decided to collect it or keep all of that dirt. So knowing how to collect is an important step in making a good vouchered specimen and process. Of course, you also want a specimen that's not too young or immature. So yeah. So good question. And do you have any recommendations or links for a favorite collection slip template? So yeah, if you go on a Mushroom Observer, either iNaturalist or Mushroom Observer are going to have templates for voucher slips. And I've got my own set that I use kind of selfishly for our own collections, and they're a little bit smaller than your average ones. But Nathan Wilson, in part of his updating of the Myco portal or the Mycology collections portal, not Myco portal, I'm sorry, Mushroom Observer, he's actually creating slips that work directly with the Mushroom Observer app and allows you to push the specimen record into an observation, so that observation automatically records your field slip number. And that's kind of a trick that iNaturalist doesn't do and probably will never do. And it's a kind of a neat trick because then we can actually connect the specimen itself in our pool, a basket of specimens, back to that Mushroom Observer observation. So it was something that Nathan Wilson and I talked about at Telluride one year, and it was a cool little development that he's starting to work on. The NAMA vouchers tended to get smudged and hard to fill out when wet. That's true. Oh, write in rain paper. Nah, thank you. Thank you for clarifying. Well, that's kind of going to be kind of expensive. I don't know a good answer to that. I think maybe write in rain paper. One thing that we thought that I was like thinking of, because like downstream, maybe having adhesive stickers for your 4A number that you don't have to, so you don't have to write the number. You can just like grab a sticker and stick it on the slip. And that way, you know, you don't have to write anything. It just stuck to the paper automatically. That was kind of one answer, but then it doesn't help you putting your name down. It just does, it helps with the where and when information. So yeah. Um, I guess try to stay dry. Here in Colorado, I don't get too wet too often. It's pretty dry here in Colorado, but I know what you mean, especially at the last NAMA foray. It was quite wet. And I guess self like specimens that are already numbered, and what you can do and what I've done in the past is just take a picture of the slip, take a picture of the specimen with the slip, take a picture of the specimen by themselves, put that all in the observation. Downstream with, I don't have to fill it out in the field. I can wait till I take the specimen downstream and fill it out later. I sometimes I get chewed out by my volunteers because I haven't filled out the slip properly. And then it's like a month or two afterwards when we're processing the specimen, and I'm like, oh, oops. So waiting to fill out the specimen slip, I don't recommend, but if it's wet and you're stuck, ideally having them pre-numbered and then having that number in the iNaturalist observation downstream allows you to link up that specimen with the observation. Because ideally that observation is going to have all of your where and when information wherever you need it. So yeah. Yeah, so write and rain notebooks, that works out really good. Good. Are lichens being vouchered at Namaforays? Yes, there are. We didn't do a lot at this last Namaforay because we didn't have any lichen people, but I think we do have a collection for this last NAMA. And then in Missouri, and then the most recent one in North Carolina, we did voucher a lot of lichen specimens. We vouchered some specimens for Colorado, but I think I'm still processing the Colorado lichens. We don't collect lichens here at the San Mitchell Herbarium of Fungi. Mostly all of the lichen specimens from Colorado are at University of Colorado Boulder with Aaron Tribb. But yeah, so fresh specimens at 4A is a goal. Microscopy would be good, especially if we could get camera and image data. And especially we can link some of that image information. If somebody's got an iNaturalist record, we could upload those all kinds of pictures to the iNaturalist records as well. What is your estimation of, this is MKR follow-up. What is your estimation of how much that especially documenting on iNatch other records that micromorphology, fresh prehydrates would help improve our vouchers? It's hit or miss. Oh, Mariah. Hey, how you doing? I think it's going to be useful going downstream, but good thing about mushrooms is that the dried specimen you can always return to get that micromorphology. Also, if you have good members or good identifiers, like if you have a Noah Siegel, he's going to be identifying everything just by look. But he's a savant like that. Not everybody's going to be able to do that. And here at Denver Botanic Gardens, pretty much every specimen we look at is going to be looked at under the microscope to a degree to sort of validate the species that we're putting on them, because we really work hard to try to create the best um, data possible. But I think at the forays, because everything has to go through in a conveyor belt like fashion, we're really just going to use the IDs based on that macro morphology. And then downstream, if researchers are really interested in validating it, that's when they can include the micromorphology in the descriptions, and then maybe use that micromorphology to overrule what was said at the NAMA foray. That's also what makes vouchers cool. But there were some specimens at this latest one where we actually, I think it was an ascomycete that we needed to look at under the microscope to really even just get it to genus. But yeah, micromorphology can be important. Many have lamented how the pipeline from amateurs to institutional fungaria is a one-way street where specimens are deposited but are inaccessible to non-academic researchers. Is discussing the Fungarium Working Group on the loan? Yes, this is exactly what I'm hoping that the Fungarium Management Working Group will do. And in my discussions with the Fungarium members, the people who are managing Fungaria, they don't really have uh, too many problems with people using the collections. The problem is because we, as a collection of institutions, we have a recognized mandate that these specimens are valuable and important for current and future science. And as a result, there is a need to preserve the specimens as much as possible. Now, what does this mean? It means essentially you want the specimens to always be in climate controlled conditions and with people who we know and have a certain amount of confidence are going to be responsible with these vouchers and these specimens because they're valuable. They're people's time, energy, sweat, and everything. And just the house them in a collection is time and energy and money and resources. So for this reason, sending them to private individuals and letting them sit in their house without adequately understanding what the specimen is in for is kind of a problem, is a bit of a challenge. Now, that's not to say that individuals can't develop relationships with institutions and develop an understanding and set in some trust around what they might be able to borrow on a loan. What we've done is Dr. Roy Holling with his wife Barbara Tears, they moved from New York Botanic Gardens to Colorado because they have family out here, and they now volunteer here at Denver Botanic Gardens. Roy is still an active mycologist, and what he wanted to do is get loans from New York Botanical Gardens of specimens that he collected, often from some foreign country, bring them here to the San Mitchel Herbarium of Fungi, which we create these interinstitutional loans so he can have access to his specimens. But more than that, what he wanted to do is be able to bring these specimens home. There was some discussion early on as to does this make sense? Should we do this? First off, we need to make sure New York Botanic Gardens is okay with it. And then the question is, are we okay with it? Are we setting a bad precedent? What Roy ended up doing is he bought an herbarium cabinet. And when it comes down to it, it's kind of hard to argue that there's going to be anybody more qualified to take care of his own collections than the guy who made the collections himself. So to a degree, there's probably arguments to be made if you are an amateur mycologist and you give your collections to an institution, but then downstream say, hey, I actually want to study these specimens. It kind of makes a certain amount of sense that, hey, these are your collections. We trust that you're going to take care of them. So let's work on some sort of system where you can borrow them, maybe look at them at your home, and then bring them back after the loan is over. So good question, Josh. I think there's a lot of room to grow on this question, and the Fungaria Management Working Group, I think, is going to be interested in discussing it. But it's going to be every institution is going to come up with their own set of rules, and it's going to be an issue that we're going to have to vet and practice on. Let's see. So on ResearchGate is an article on collection methods. Any thoughts? Okay. Let me click on that. The collected guidelines. Okay, cool. Oh yeah, Bark Bike, and this is a 2010 paper. Ooh, this is good. Thank you for showing this to me. I'm going to download this. And yeah, I think this is going to be good and important for us understanding guidelines on collecting and then using those guidelines when we create a broader set of instructions moving forward. And actually, the collection guidelines are from a chapter in a book, Manual on Field Recording Techniques and Protocols for All Types of Biodiversity Inventories. Ah, yes, that ABC thing. Okay, good. Yeah. I actually probably even have that somewhere. And there's also another book out there on collecting macrofungal collections. So, but other than that, I don't know if I'm supposed to sign off on everything or not. Trent, are you still here?
Yes, yes. Hello.
Hello. Yeah, I feel like I've gone
Back
to
this week.
everybody's questions. Yeah.
Yeah, I think you did pretty good. You got everybody's questions answered. So I guess it's time to sign off. And I want to just officially and gratefully thank you for coming on and spending like an hour and a half with us. Sharing all your knowledge on the subject. So thanks so much for doing this for us tonight.
Thank you very much. I'm really excited to talk about this topic, and I'm glad everybody was able to join. And I really appreciate paying attention to this issue and the attention given to it. So thank you for giving me the opportunity.
Okay. Well,
To,
great. Good night, everybody. And we'll put this up on YouTube next week. So if you want to watch it, we'll be there. Thank you. Good night.
Great. Bye bye.